Hey, how are you?
Good, how are you?
I'm good, I'm good.
All right. Let's do. So, did you get the zip package that I sent you last time?
Oh, yes. Let me take a look at it now. Okay. Here, let me just download it.
I was hoping we can go over the biology section first, and then if
Yep,
we have time, we can do the chem phys
got it.
section. Yeah. I just did the practice last weekend.
Let's see. Passage 10, okay.
It was, I think it was passage 5, 6, 7.
Mm-hmm.
Yeah, like the later ones that I had a few questions from like each of the passages.
Mm-hmm.
Yeah.
Let me extract. Okay. Calorie restriction, cystic fibrosis, Ebola virus. Okay, so I see the calorie restriction one, and let's see. The best experimental method to analyze the effect of TDH2 gene deletion on the rate of histone acetylation.
Mm-hmm.
Um, it should be the one that lets you see the change in proteins.
Right. So I eliminated C and D because I knew it wasn't gonna be those two because those deals with RNA. And I was just kind of confused on like the gene deletion part because so I was really debating between the two because histoacetylation is like protein related, but then I also talked about gene deletion, which is like DNA related. So I got mixed up, and I don't know
Um,
like yeah.
okay. So I mean, they're talking about like anytime they're talking about histones and stuff, they're talking about expression of a gene.
Okay.
Like that is like have you ever heard of epigenetics?
Yeah.
Okay, so like that's become much more of like an important thing, um, involving like whether or not a gene is expressed or not.
Mm-hmm.
So if they wanted to see the effect of that gene deletion on the rate of histone acetylation, right?
Mm-hmm.
And they're comparing wild type and delta TDH2. So I guess that's the one that has that deletion.
Yeah.
So wild type, I suppose, doesn't have a deletion, and the delta TDH2 has deletion.
Mm-hmm.
But we're trying to see the rate, or we're trying to see like the difference in histone acetylation, which is something like if it's acetylated, the histones get further apart, so they're less condensed,
Yeah.
and transcription factors can bind to that DNA and cause the RNA to be transcribed.
Yeah.
And they're transcribed and later translated into the protein, right?
Mm-hmm.
So we're going to be dealing with a protein, whether a protein is expressed or not.
All right. So the expression of the protein. Right. Okay. Okay.
Mm-hmm.
Okay. I was just confused because I was like, if they deleted the gene, then obviously it's not gonna get expressed. But
Yeah,
not
but they're
necessarily, right?
comparing the wild type to the deletion, right?
Yeah.
And so one part will be the gene is completely deleted, the other one it's not deleted. But
Yeah.
since they talk about uh, the rate of histone acetylation, that is something that you know it is something that affects like the expression of the gene, and therefore is a protein kind of product.
Mm-hmm. Yeah, that makes sense now. Okay. I was just confused. Okay.
Mm-hmm.
Okay, next question is genetics related.
Cystic fibrosis.
Yeah.
Okay, so let's see. Female. Okay, so all right. Let's see what this stuff is. Okay, so this is If a female heterozygous for this bears a child fathered by a male that's heterozygous for another allele, what is the probability that the child will be homozygous for that one, given that neither parent... Okay, let me take a look. So, cystic fibrosis, all right. It's in the autosome. Oh, okay. That could help you. But wait, let's see. Okay. Most common mutation of the F508 allele results in, okay, homozygous for this means that they have cystic fibrosis, okay? And then, okay. And then let's see. Now, if they're heterozygous for it, okay, they have better resistance to typhoid, okay. Let's see. I'm trying to look for the other one. Okay, I guess it's not there. So let's see. So they're just introducing this in this question. So they're saying that there's another allele, and a female is heterozygous for it.
Mm.
Okay, and then the male is heterozygous for the one that they talk about. What's the probability that the child would be homozygous for that allele, given that neither... Oh, okay. So if you do like a Punnett square kind of thing, right? Because they would have to be homozygous for that G542X allele. And you basically just have two that are heterozygous. Like, well, you have one person... Okay, so let's see. The female is heterozygous for G542X. So I'm just going to use, like, capital A, little a. And then the male basically doesn't have any of that. Wait. Doesn't have any of the G542X allele. Probability that the child would be homozygous for that. I mean, so the father doesn't have that at all.
Wait, where did you see that?
Well, all they're saying is that the father has the F508 allele.
Yeah.
And they're asking what's the probability that the child would be homozygous for the G542X allele.
So are we supposed to assume that the father doesn't have the...
Yeah, because it doesn't tell us anything about the father with that allele. All it says is that the female has the G thing and is heterozygous for it, and the father has the F1 and is heterozygous for that.
Oh, okay.
So, and they're asking for the genotype, or rather the probability that the child would have, like, be homozygous for the G1. And since the father doesn't have any of the G1, there's a 0% chance.
Oh, okay, I see.
Yeah. If the father was heterozygous for the G thing, like the mother, then it would just be like a heterozygous cross, which will give you a 25% chance for her.
Yeah.
Okay,
exactly.
Yeah, and yeah, but the father doesn't have any of it, so.
Okay. Yeah, I mean, this question I think threw me off because I was just like, how can it be 0? Or like, I think I just like was overthinking it. Yeah, so I don't know. So I think it's...
Yeah, because they're asking for a specific thing, like homozygous for the G allele.
Yeah.
So that's a genotype question, and that could be answered like pretty specifically. It would have been harder if they asked what's the probability that the child would have cystic fibrosis, because that's more of a phenotype thing.
But that is the question, though. does the child have CF?
Well, they're just asking if the child is homozygous for that G allele.
Oh, right.
Yeah. Because if they're asking for cystic fibrosis, it could be the 25% one, right?
Yeah.
Because they don't really tell us about what the G thing does, but they tell us that the F thing, like if you're homozygous for the F one, the F allele,
Mm-hmm.
you're going to have cystic fibrosis.
Right.
So if the G allele is also like that, then there's whatever 25%. Then cystic fibrosis could be something. But since they're asking for a specific genotype, then it makes it easier.
Yeah, for sure. Do you feel like sometimes the exam just like makes questions that are like kind of obvious but like make you overthink? Or
Yeah, yeah, yeah,
okay. All right.
yeah.
All right.
It's designed to psych you out.
Okay. All right. Next question.
Because I'm looking at like the word autosome,
Yeah.
and I'm looking at the frequency of that F allele, which makes you
Hardy
think of
Weinberg.
exactly.
For sure.
So there's a couple of things that could lead you in a different direction.
Yeah. So just to check,
Yeah,
so autosome means it's like not on the sex chromosome, right?
correct.
So for example, if I do the cross, I don't need to do like the XY thing, right?
Yeah.
Okay, got it. All right. All right. So the next question is related to hormone.
All right. Scientists are hopeful that NPY, which is neuropeptide Y. Okay. All right, and then they give us LH levels in the. I'm just looking at table one.
Mm-hmm.
Um, so first of all, in the table one, I'm only going to pay attention to the ones with asterisks.
Mm-hmm.
So saline NPY alone, I don't care about. I care about GNRH alone and GNRH with NPY.
Mm-hmm.
And there is, statistically significant. Or wait, asterisks say, all right, for GNRH alone, significantly greater than LH levels in the saline sample. Okay, and then for the double asterisk, okay, they're saying significantly greater than LH levels in just with. Okay, well, yeah. So they're just saying that these are significant. So okay, now back to the question. Scientists are hopeful that NPY can be used in combination with GnRH to treat certain cases of female infertility. Individuals with a deficiency in what receptor system would be most likely to benefit from such a treatment? Okay. So it's not going to be NPY or LH and NPY because those things like NPY, like, let's see. Okay. LH levels, and let me just connect that to infertility or fertility. Um, okay, let's see. So, deficiency in what receptor system would likely benefit from such a treatment? I think it's almost the same thing where you look at the table one. So
Mm-hmm.
so basically like NPY really enhances um like like it enhances like or the addition of it with GnRH enhances how the GnRH works alone, which works down the line for production of like LH and stuff like that. So
So so deficiency means lack of, right? In this case, or like not as much of.
correct, yeah.
So I thought that this question, the answer is LH
Mm-hmm.
because so GnRH increases the level of LH, right?
Um. Yeah.
So if a person was deficient in LH, which means this person doesn't have LH, and if you add a treatment that has MPY and GnRH, then that will enhance the level of LH, which the person doesn't have. So I thought in that case, the person will benefit the most because if you don't have GnRH, then like MPY can't act on it, can't modulate it. Do you know what I mean?
Um. Yeah. So I think the key here um is okay. So they're using LH as just like a thing to measure, like kind of like a dependent variable here. So they're using
Mm-hmm.
LH to measure that, like as a whatever dependent variable to see
Mm-hmm.
like how it changes, and in the presence of saline, or just NPY, or just GnRH, or GnRH and NPY, right? And then they're
Mm-hmm.
saying in the question that when you use NPY with GnRH, it can work to help certain cases of female infertility. So, individuals with a deficiency in what receptor system? I think the system part could help because,
Oh,
yeah,
okay. In what sense? System.
because so okay, like tell me what you know about like the hypothalamus and the pituitary.
Mm-hmm.
Yeah,
uh, so I think GnRH is produced by the hypothalamus,
yeah.
and
Basically,
then
all of these releasing hormones
yeah,
are yeah,
produced by the hypothalamus. So that stimulates the release of LH from the anterior pituitary gland.
mm-hmm, yep, yep,
And then that LH will stimulate like I think ovulation or whatever and
mm-hmm.
progesterone or something. Yeah.
Yeah, it can lead to yeah, it could lead to a couple of things. But predominantly, what we're thinking of in this case, I see in the first paragraph, they're saying that um, LH stimulates sex hormone production by the gonads
Mm-hmm.
and triggers ovulation, right?
Mm-hmm.
And let me see if it says okay. Okay, I thought they were going to say something about. All right, well, okay. So I thought they were going to say something about the creation of like the ova and stuff. Um, but I guess they mean in the because I was looking at female infertility.
Mm-hmm.
So yeah, so maybe it's not necessarily the creation of the ova, but just the fact that LH triggers ovulation, and you need ovulation in order to be fertile because that is what lets the ova like be released and get be ready for fertilization.
Mm-hmm.
So okay. So let's see. So LH versus um, GnRH. The deficiency in what receptor system? Receptor system. All right, so there's hypothalamic factors. Okay.
Like I can see that both deficiency in GnRH and LH can benefit, but I'm just not sure why GnRH is most likely to benefit from the treatment.
So I think all right. So I think maybe two things. So the GnRH alone produces significantly greater LH levels than just getting the saline.
Mm-hmm.
Okay, so like, and since GnRH comes before the release of luteinizing hormone,
Mm-hmm.
so okay, so they're saying a deficiency in which receptor hormone would um, benefit from such a treatment. So, okay, let me see.
Actually, I think I can sort of justify this answer now.
I mean, I think basically they're talking about. I think when they say receptor system, right?
Mm-hmm.
And they're talking about in the second paragraph, like
Mm-hmm.
just how the hypothalamus basically has this like system with neurons
Yep.
that basically work um, to release LH, I believe.
Mm-hmm.
So okay, let me see. So why isn't it LH and why is it
I think I
GnRH?
have a theory.
Yeah.
Okay, so basically, I think most likely to benefit. So I'm thinking so it's related to the outcome. So let's say the outcome is infertility.
Mm-hmm.
So if a person doesn't have LH, but the person has GnRH, then technically the person should still be able to produce LH because after you add the treatment and you stimulate GnRH, then you can make luteinizing hormone.
Mm-hmm.
So so then it could still help the person become fertile. But if the person doesn't have GnRH, then the person will likely to not make LH, which means the person will be infertile. So then if you add it, it will like benefit more.
Yeah. I was thinking of it in the sense of receptor system because the GnRH is what causes things like the LH to be basically like made, and then that works, elsewhere. Like it says, GnRH is a potent stimulator of pituitary LH production and release. So let's
Mm-hmm.
say, so imagine like we're gonna break something, right? Like let's say we break um, the, or let's say that someone had a deficiency in LH. You can
Mm-hmm.
still fix that by adding GnRH because that's what stimulates the LH in the first place.
Mm-hmm.
But if you had a deficiency in GnRH, since it's like upstream kind
Yeah.
of like everything else, like that is that is what can be enhanced by using NPY and GnRH.
Mm-hmm.
So like
Yeah. I
yeah, if
agree.
if I come up with a better idea, I'll tell you because I'm looking at the receptor system, so I'm just thinking like just the fact that the hypothalamus uses like basically neurosecretory neurons to secrete stuff into the anterior into the pituitary glands.
Mm-hmm.
So that
Yeah.
is
No, that
a
makes sense.
yeah, that's like a
That's
receptor system.
yeah, that's kind of what I'm thinking about. But yeah,
Mm-hmm.
I agree.
Yeah, if I
But
see a
it's just
better
I'm not sure if that's the level of thinking that I need to go to because I on the test, I will for sure not think like that or like that level of like detail. So I don't know.
Well, like, you know, the hypothalamus, that's like the relay center,
Yeah.
right? It takes in information from your brain, and it, you know, it would releasing hormones and stuff, which would cause like effects downstream.
Yeah.
But that's like the, you know, where everything basically happens.
Yeah, for sure.
Let's try, yeah. But yeah, if I see like a better explanation for that, or come up with one, I will tell you.
Okay, sounds good.
Yeah.
Let's see.
Let's see what's next. Ebola virus.
Yeah. So the next passage is primarily the figure that I'm not sure how to interpret.
Yeah, so that's like the hard stuff when it comes to the BB section. Interpreting like, yeah. Okay, so it seems like okay, so percent of total cells infected and then introduced genes on the x-axis. Okay, so for wild type, right, you basically have that amount of infected cells, like whatever, that's like 27% or whatever.
Mm-hmm.
All right, now let's say we introduce some bacterial cells.
Mm-hmm.
So I guess that means that almost none of the cells are infected.
Mm-hmm.
Okay. Now, in some okay. Sorry. So bacterial cat B and cat L, that's in a mutant that doesn't have cat B, but has cat L. So let's see what those do. So cat.
Wait.
Oh yeah,
Is
sorry.
are you talking about the first 3 columns after the wild type?
Yeah.
Wait, can you say that again? What does the top part mean? Like the
Oh, that like top bracket.
yeah.
That just means that's in so mutants that don't have the ability to make cat B, but has the ability to make cat L.
Wait, hold on.
Mm-hmm.
Um. Okay. Okay. So they're talking about the cell, right? Is it
They're talking about the. Oh wait, sorry. What are you asking? Yeah, the cell. Mm-hmm.
so so basically they made the cell so that it's unable to make cat B, but
Mm-hmm.
it can make cat L, right?
Yeah.
Okay, but then so at the bottom it says it introduces the gene. So because it doesn't have the ability to make cat B, but then we're introducing the gene to make cat B. Is that what it's saying?
Mm-hmm.
Okay.
Yeah. So basically, as is, like bacterial introduction of bacterial genes don't really mean anything. That's kind of just like treated as a control.
Mm-hmm.
Okay. So, all right. So basically, like, yeah, the control and the introduction of cat L doesn't really do anything. And it should make sense because the introduction of bacterial genes doesn't do anything, and cat L, it already can make cat L. So no real changes there. However, if you introduce cat B and it can't make cat B itself, you see a dramatic increase in the total number of cells infected.
Mm-hmm.
Okay. All right, now we have the next bracket, and this is like a mutant that is unable to produce cat B and is also unable to produce cat L.
Mm-hmm.
All right. So introduction bacterial, again, nothing, treated as a control. Introduction of cat B only, it goes up like a little bit.
Mm-hmm.
Introduction of cat L only, doesn't really change it. Introduction of both cat B and cat L makes it go up substantially.
Okay. I see. Okay. So I'm thinking
Mm-hmm.
from the previous column
Yeah.
where it's like you can make cat B but not cat L, and when you introduce cat B, the infected cells dramatically increase. Is
Wait,
it
you're
because
saying in which one?
the s- the first one after the wall type?
So with the one that doesn't isn't does so not able to produce cat B, able to produce cat L. Okay.
Yeah. So when you introduce cat B, because it has the ability to make cat L, so it's gonna make cat L, right? So that's consistent with the second part, right?
Yeah, it seems like both of them like have some type of cooperative, synergistic effect.
Okay, got it. Okay, I see. So then, okay, so one more question. So with the brackets and the cat B notation with the plus minus sign, what does it mean? Like, does it always mean like, oh, this cell can't make
Uh,
something?
like, uh, like cat B negative slash
Yeah,
negative.
yeah, yeah.
Yeah, yeah, it's a knockout one.
Of the gene that makes cat B, for
Mm-hmm.
example. Okay, okay, hold on. I need to write this down.
Yeah.
Uh, knockout of gene that. Okay, because I'm never really sure what it means.
Mm-hmm.
Okay. Okay. So let's see. Based on is the cat L sub expression? Okay, so based on the figure, the answer should be no.
Yeah, it should be
So
no because
then,
cat L alone isn't enough.
right. So then, based on C and D, let's see, because affected. So D is wrong because both cat B and cat L doesn't express it better than cat B.
Yeah, because it says infect cells expressing both better than it expresses cat B but not cat L, which means that it's cat B and cat L work just as well as just cat L.
Wait. Hold on.
Mm-hmm.
um. So it's so answer D is saying that both cat B and cat L is better than it infects cells expressing cat B and not cat L.
Mm-hmm.
Wait, that's true.
So that's saying that cat B plus cat L works better than just cat B. That's
Yeah,
true.
that's true, right?
Yeah, but it says but not cat L. So
Uh,
see if you can.
uh,
Yeah.
wait, hold on. This is confusing. Wait, what does but not cat L mean?
Here, let me do this. Let me do like a share. Oh wait, can you enable share
Oh,
screen sharing?
okay, try it now.
Okay. So you see this like my Microsoft whiteboard thing?
Yep.
Okay. So this is what it's saying. It's saying cat B plus cat L basically works better than cat B. That's
Mm-hmm.
true.
Mm-hmm.
However, the second part says, but not cat L, which means cat B plus cat L is not better than just cat L.
Right. That is not true.
Correct. And you see how, like, yeah, this is what I wanted to work on, like, before, like, going through a passage and writing it in this type of shorthand, which helps you decipher, like, what they're saying, because, there's tons of, like, double negatives and things like that. So,
Mm-hmm.
yeah, that's not true. So that's why D is not it.
Right. Yes. Okay. I
Whereas,
think,
yeah, so,
yeah.
so that's D. But C, what C is saying is that um, cat, let's see, cat B minus plus cat L is not better, so is equivalent to cat B minus plus, or rather, minus whatever, cat L. And, let's see if that's true. So, cat B, without cat B, and you have cat L, it's basically, or here, I'll do this.
Yeah.
So, okay. So, cat B minus plus cat L is this over here,
Mm-hmm.
right? And then
Yeah.
cat B minus without cat L is that.
Mm-hmm.
Oh, wait. Sorry, not that. Sorry, because that means that cat L is still there, and this is without cat L. So that's going to be like this one. So either that. So basically that, I guess.
Yes. Okay,
Because
hold on. Let me read it.
yeah, because this is something that doesn't express cat B,
Mm-hmm.
but you're introducing cat L to it. So it's gonna
Correct.
be. Wait, actually, hold on. So okay, let's actually just use this over here.
Okay.
So, because all right, no cat B, no cat B, right? No cat B, right? And since this is an introduction of cat L and this is not cat L, we're gonna be able to compare the two. So cat B, so this plus that is gonna be this over here,
Mm-hmm.
and then this over here is without cat L added is just gonna be the basic version of that. So bacterial,
Mm-hmm.
and they're equivalent, right?
Yeah.
Yeah, and that's what the statement. That's what C says. They're saying that. Um, so like to make sense of this because it's a lot of words and stuff like VSV EGP does not infect cat B minus cells expressing cat L better than it infects cat B minus cells not expressing cat L.
Mm.
So so I made sense of that by writing cat B minus plus cat L, right?
Mm-hmm.
Equals, right? Cat
Yep.
B minus without cat L. And then this is where I found it in the whatever figure one.
Mm-hmm. I see. Yeah, so first of all, I did not even look at the right column. That's problem to
Oh,
start
the right
with.
column, like the ones that are under here.
Yeah, no, it's like before today, I didn't look at the right one. So I feel like
When you
that
say the right
probably.
column, you mean these?
Yeah, like I wasn't able to identify which column I'm supposed to look at.
Oh, okay.
So yeah. But after we broke it down, like I get it now.
Yeah. Originally, like for this, you could also use that because cat B minus, cat
Yeah.
L plus, that is going to be the same. So both of these things can be that. However,
Mm-hmm.
to not have cat L means that you can't do this. You can't do this because that has cat L, and you
Yeah,
have to use this, which doesn't have cat L.
for sure.
Yeah.
Yeah.
So yeah. So these are just ways of taking like the verbose like language that they use and like putting it into like a shorthand, which makes it easier to like understand and comprehend. And then yeah, when we went over the figure one, like that's how to like look at it.
Yeah, this is definitely the one of the trickier question types, I think.
Yeah, and the overall like thing that you should get from this, from the entire figure, is that cat B plus cat L is basically, you know, greater than um like if it's natural, I guess, is greater than wild type, which is greater than cat B plus cat L like added. Because I'm looking at the largest one over here, which has cat L natural and then cat B added.
Right.
And then the next highest peak is just the wild type. And then the second, or the third highest or whatever is without these like naturally there, but the genes added afterwards. Yeah, in terms of like infectivity or whatever.
Yeah, makes sense.
Yeah, so I use a lot of like less than, greater than symbols and stuff.
Right. No, yeah, definitely.
So make sense?
Yeah, makes sense. Okay, I'm gonna try to do that. I'm gonna try to use like I use so when I draw my like diagrams or whatever on my paper, usually I do because I think some of the questions they ask a lot about like, oh, this thing inhibits this or this thing stimulates this. So I use plus and minus sign for that, but I've never used like greater or like less than sign. So I feel
Oh
like I
yeah,
can try.
yeah. If we're inhibiting stuff, I guess you'll see what I use when I see it, when I do it. I usually draw like an arrow, um, and either like a flat end to that arrow or like I cross out the arrow. But yeah, let's see the next one. So the next one says, based on the passage, does optimal VSV EGP infection in vitro require cat B, cat L, or both?
Okay, wait. I think
Oh, so this is like from before, yeah. So this is like, so see how I did this over here? So yeah, or I think you knew that from like an earlier question, or maybe yeah. I think you knew that. Like
now I do.
mm-hmm.
Yeah. Now I understand. But
Yeah.
yeah.
Yeah,
Before
yeah. What were you saying?
no, like before I didn't understand how to read the diagram and I had problems. But now that I see it,
yeah.
not I get it now.
Okay. Cool.
Okay. Yeah.
All right, let's see the next one. All right. Given that secretory lysosomes form normally in the melanocytes and CTLs, which are cytotoxic T lymphocytes, which are also known as I think the same as natural or natural killer, whatever, something like that.
Killer T cells.
Killer T cells.
Cytotoxic T cells equal killer T cells.
Okay. So, but natural killer cells are different, I think, right?
Yeah, they're like part of the non-specific, I think.
Got it. Okay. Okay, so so secretory lysosomes form naturally in the melanocytes and CTLs of ashen and dilute mice. The data in Table 1 best support the conclusion that melanosome secretion and lytic granule secretion differ in the secretion of lytic differ in that the secretion of lytic granules does not require. Okay, So let's see. So I'm gonna look at the passage for a sec. Okay, so all right, these both use modified lysosomes, secretory lysosomes. Okay. All right, CTLs. Okay, so here I'll write this out here. So I'll say this. Let's see. Melanocytes. Okay, so I'll just write CTLs plus melanocytes. I'm just gonna use capital M. Um, they both cause. All right, they both use secretory lysosomes. Okay.
Mm-hmm.
All right. Now, how do they differ? So CTLs use ones that are called, okay. So they have lytic granules that secrete cytolytic proteins. Okay. So lytic granules. Here, I'll go closer here. Which secrete cytolytic proteins. Whereas the melanocytes use melanosomes,
Mm-hmm.
which transport melanin pigments.
Yeah. By the microtubules.
Yeah. Generally speaking, yeah. I'll I guess come back to that if I need. Let's see. Then something whatever. Oh, okay. Since this thing has Rab and stuff. All right. Let's see. Oh wait. Okay. Wait. Transport it. Okay. Okay. They transport it and then they release it.
Mm-hmm.
Okay. Now. Okay. Rab24A protein on the melanosome membrane facilitates this transfer by interacting with something which binds. Okay. So there's some crap happening here. So let me write it down next to it. So basically, what's happening here is that. All right, how should I do this? Let's see. So, um,
So how I visualized it is like melanosome interacts with the melanophilin protein, and that protein binds to the myosin VA.
mm-hmm.
That's kind of like how I understood. Um,
Okay, so I drew something like this. The melanosome on the membrane of it is the Rab27A, which basically interacts with this melano something filin protein. which binds. So okay, I'll say binds to the myosin VA, which is a motor protein on the microfilaments.
Mm-hmm.
So okay, now. All right. I was gonna skip it, but I guess I'll just do the microfilament, microtubule stuff. So let's see. I'll do something like this. Melanosomes. Let's see.
I just kind of see the melanosome going along this thing called the microtubule.
Mm-hmm.
I just kind of imagine the melanosome traveling along the microtubule, pretty
Mm-hmm.
much.
Oh, damn it.
Yeah, that's kind of roughly how I pictured it.
Mm-hmm.
But the question is still very confusing.
Okay, so let's see. So I wrote melanosome. In addition to this, it moves along the microtubule towards
Mm-hmm.
the cell periphery,
Mm-hmm.
where it gets transferred to the microfilaments, and once it's there, it releases its melanin.
Yeah.
Okay. Now let's see. People with this syndrome have hypopigmented skin. All right. So sometimes they're not always associated with immunodeficiency, resulting from a defect in the killing function of CTLs. Okay, characteristics of two mutant mouse lines are used as models for Griscelli syndrome, as shown in Table 1. Okay, so let's see. So basically, all right, Ashen is the mutation, and dilute. Okay, killing by CTLs. So that's that. You know, like the immunodeficiency part where the CTLs aren't. Wait. Killing by CTLs, protein inactivated. Okay, partial albinism. Okay, and then okay, this I see. So this mutation causes this to inactivate, and it results in, I guess, the CTLs not being able to kill anymore.
Mm-hmm.
And you get partial albinism, and then this mutation causes the myosin VA to be inactivated, which keeps the CTL functioning, and you still get partial albinism. So let's see. So basically, let's see. RAB. Okay, so when RAB is messed up, basically you don't have the melanosome transfer. Wait. Yeah, you don't have it transfer to the microfilament, I suppose.
Mm-hmm.
Okay. Now, when the myosin VA is messed up, you don't have it attached to the microfilament, I guess. Okay, let me see. What are they asking? Given that secretory lysosomes form normally in the melanocytes and CTLs of ashen and dilute mice, okay, the data in Table 1 best support the conclusion that melanosome secretion and lytic granule secretion differ in the secretion of lytic granules does not require. So if myosin VA is messed up, so okay, they're basically saying that lytic granule secretion, right, is this over here,
Yeah.
and that is what causes the CTL to work to destroy, to kill, right?
Mm.
So if RAB is messed up, this thing is messed up.
Oh, wait. I think I messed it up. Okay. Okay. So based on Table 1,
Yeah.
okay, let's look at, so without RAB 27A, then you don't get CTL.
Correct.
So that means you need it, right?
Yeah.
Oh, okay. Okay, so then myosin, okay, without myosin, you still get the CTL.
Yeah.
So then it doesn't require.
Yeah.
Okay. All right. Oh my god, this is so complicated.
So I like, if you look at like what I drew out here, and then based on RAB and myosin, and that CTLs, they function through lytic granules,
Mm-hmm.
right? So, and they're asking about like what does the lytic granule secretion require, right? So think about like CTL function like is proportional to its lytic granules,
Mm-hmm.
right?
Mm-hmm.
And so if you can like equate the two, so like and basically like, so RAB 27A, when that is inactivated, the CTL function is inactivated.
Mm-hmm.
So that's something that is required.
Yeah, correct.
Myosin VA inactivated, enable like the lytic, the CTL function is still there, so the lytic granules are still there. So it doesn't need myosin VA.
Mm-hmm. Yeah, I see that.
And as for microtubules and fusion of these secretory lysosomes to the plasma membrane,
you can't tell, right? Because it's not tested.
Yeah, it's not tested. Yeah.
Yeah. At first, I thought I could get the
you know
answer.
what? It would be tested if there was anything without partial albinism, because the microtubules and this part are what enable the melanin to be released, causing someone to like if the melanin is released, then they have melanin, the dark pigment, and they wouldn't have partial albinism.
Right.
So since both of these mutants have partial albinism, right? That means that, well, I mean, they're talking about the lytic granules. So actually this question, like microtubules and this part, wouldn't even relate. But they could ask you about microtubules and stuff if we had, a table that had someone without partial albinism.
Yeah, I can see that.
Yeah, like let's say I gave you a question that said that um, to support the conclusion that melanin release or secretion, um, I guess, differ in that the secretion of melanin, um, melanosomes or whatever, does not require like these guys, then it would be something like microtubules. But I'm just like using that as an example of how these things relate to this part.
Mm.
Yeah.
Yeah, I think I was confused
Mm-hmm.
because from the second paragraph where they describe the differences, I
Mm-hmm.
thought those were the only differences. So initially, when I saw this question,
Mm-hmm.
I was like, oh, I was gonna pick A because A was talked about related to the melanocytes and not the CETLs.
Mm-hmm.
But then option B, which also was related. So then I had to look at table 1 in order to figure out the answer.
Yeah. And they tell you, you know, to look at table 1.
Yeah.
So yeah, first I was gonna look at table 1, but then I was like, all right, I don't know what this is or this is. So then I
Mm-hmm.
went and actually looked at the passage, and you know, I drew this out, and then I was reading the second paragraph, and you know, just to get like the basics to fill this art out, I knew that the microtubule and microfilament part wasn't going to really be that important. Um, but I ended up, you know, writing about it anyway.
Right, right.
Because
Well,
yeah, they might
the
ask you another question about it or something, right?
yeah, the microfilament. Well, they asked about microtubule and microfilament in 2 of the other questions, but
Oh, okay.
yeah. But yeah, I think
Yeah,
I
those
was
have more to do with the albinism aspect
mm,
because
yeah,
they're crucial for the melanin to be released.
interesting. Yeah. I think I was confused because after they said so the first after the first sentence of the second paragraph, they
Yeah.
just went on to talk about melanocytes. So I was like, oh, okay, so that's everything I need to know about the
Mm-hmm.
cytotoxic. But I think they just omitted that part on purpose, probably,
Mm-hmm.
because otherwise it'd be too easy if they said like, oh, um, the mechanism of CTL also kind of relates to melanocytes.
Mm-hmm.
So I was very confused by that, but
Mm-hmm.
I yeah. But based on table 1, I think I get it now.
Mm-hmm.
Okay. All right.
Yeah. One second.
I have one. I think yeah. Just one last question from this.
Yeah.
Just the last question from the BB section.
Okay. FOXP3 X-linked gene. So what does that mean?
Oh, which one? Oh, X-linked.
Yeah.
That means it's linked to the X chromosome,
Yeah.
or it's on the X chromosome.
Yeah. I guess it doesn't say if it's recessive or not. Okay, let's see. Female mouse born with 2 wild type FOXP alleles. Okay. What is the minimum number of inactivating recessive mutations that might be sufficient in the FOXP3 alleles in mammary epithelium to significantly increase the likelihood that this mouse will develop mammary tumors? Okay, that's a lot. Let me make sense of it. So, okay. Let's see. X-linked, okay. 60% of female FOXP3 SF slash positive. So that means that they have one inactivating Skirfin SF allele and one wild type allele of the FOXP3 gene. Okay. Okay. So okay, so they spontaneously develop mammary tumors by 2 years of age. All right. Now, okay, so wild type FOXP3 transcripts are easily detected in non-cancerous mammary epithelium from female, okay, but are undetectable in cancerous ones from, okay. Okay. All right. Let me see if I can make sense of this. So, it doesn't say anything about. Wait. Okay, 60%. Okay. Which carry that and that. Develop that by age 2. Okay. Let me see what I can do. I'll do like 60% of, I don't know, my pen's acting up. Okay. Of Fox P3 SPF slash plus. Oh, yeah, it doesn't say if it's cancerous or not. So let's see. Mammary tumors by age 2. All right. Now, non-cancerous. Okay. Undetectable and cancerous. Okay. So cancerous with a C, non-cancerous with an NC. So I'll just write cancerous, undetectable, non-cancerous, detectable. And I guess in the middle, I'll just put wild type in case. All right. In female, okay, the wild type FOXP3 is carried on the inactivated X chromosome in 15% of non-cancerous mammary, about 100% of okay. So geez, okay. So
that means if the cell has cancer, then the allele will be on the inactivated X chromosome. Right?
Yeah, in 15% of non-cancerous,
right.
But 100%. Okay, so okay. Let me use another color. So for the non-cancerous ones, 15% is on the inactive X chromosome. For the cancerous ones, 100%, I guess. All right. Female, okay, cancerous mammary epithelium has 8 to 12-fold more mRNA for the transmembrane cell surface receptor ERB-B2 than does non-cancerous. Okay. So also 8 to 12 times more whatever ERB-B2, okay. FOXP3 binds specific sequences in the ERB-B2 promoter, and deletion of those binding sites increases expression from the promoter in the FOXP3 expressing gene. Okay, so I guess FOXP3 acts like as a repressor.
Right. Yeah, exactly. Yes.
Okay. Okay, and the growth rate of mammary tumor cells in the culture and the ability of these cells to cause death when injected into mice correlate inversely with the level of FOXP3 expression. So that kind of works with the repressor part. So let's see. tumor growth rate, and inversely with whatever, cell death, I guess. I'll just write like apoptosis. Okay. All right, now let's see. Female mouse born with 2 wild-type FOXP3 alleles. Okay. What is the minimum number of inactivating recessive mutations that might be sufficient in the FOXP3 alleles in mammary epithelium to significantly increase the likelihood that this mouse will develop mammary tumors? All right, so wild type, okay. Minimum number of inactivating recessive mutations. So wait, scurfin, okay, is the inactivating one. %60 have one inactivating allele there. Okay.
Where does the recessive part come in?
Inactivating recessive mutations. X-linked. Wait, what is the minimum number of inactivating recessive mutations that might be sufficient in the FOXP3 alleles in mammary epithelium? To significantly increase the likelihood that this mouse will develop mammary tumors. Okay, so if this is recessive, you're going to need 2 of them. You're going to need it on both in order for it to actually work.
Wait, how so?
Okay. Oh, wait. Okay, so 60% have... Wait. 1 inactivating... Okay, so... Alright. 2 of these. So they're asking about a mutation that would get rid of this inactivating scurfin allele, I believe.
Get rid of?
Yeah.
Because it's recessive?
So, okay, let's see. So, 60% of them have 1 inactivating SF allele. And 60% of them with just that 1 allele get tumors, right?
Mm-hmm.
Okay.
Wait, actually, I'm not really understanding this.
Oh, yeah, okay. So, yeah?
So it says that 60% of female mice which carry 1 activating SF allele. So what does it mean by carrying 1 inactivating allele? Like, how many alleles are there?
Oh, like there should be like 4 total.
4 alleles?
Yeah, like on like one X chromosome, um, it'll be like an allele for SF and a wild type plus allele.
Hmm.
So okay, so let me see if I could do this. Maybe a
Like, I
little.
guess I just never understood, like, alleles or whatever. Like,
Mm-hmm.
what is this? Like, is there a possibility that there could be like two inactivating alleles? Like, I'm just not understanding. Like,
Yeah, yeah. So okay, a female mouse. Okay, so X linked. Okay, and they have two X chromosomes. So I was thinking. So these are all. All right, wait. See. So okay, this has an SF allele and one. So it has one SF allele and one wild type allele. So okay, so now in a female mouse born with two wild type FOXP3 alleles, so that's a mouse that is basically FOXP3 plus plus.
oh yeah, yeah. Oh, okay, okay. Yes, yes, yes. Okay, I do see that. But so what what does the plus slash plus mean? Like, what does it indicate?
So each of these indicates like all right, if we go back to like that,
Mm-hmm.
like this is one allele and then there's a slash and then that's the other allele,
Mm-hmm.
right?
Wait, what's a allele? Oh my god, I'm getting so confused.
Oh yeah, so allele is just essentially it's a place where a gene is, I suppose. All right, that's a bad definition, but it's like let's see. So okay, how about this? So let's say like where is that? So on this chromosome, like at this location, is an allele. Oops, allele. Jeez. Allele for let's say like hair color,
Mm-hmm.
and let's say that like A stands for like I don't know brown, and little a stands for like I don't know like blonde or something.
Mm-hmm.
Um, so each of these are like this is an allele, this is an allele, right?
Mm-hmm.
So also like when you're dealing with Punnett squares, right? Like let's
Mm-hmm.
say like the okay, let's say that capital A is brown, little a is blonde,
Mm-hmm.
and like capital A is dominant.
Mm-hmm.
So let's say we mix like a woman with capital A, little a, which is brown phenotype
Yeah.
but heterozygous, right?
Mm-hmm.
And then whatever, a male with with that as well. Then like
Mm-hmm.
it's basically like this. So basically like 75% will have brown hair and 25% will have blonde hair.
Yeah.
Yeah. So an allele is just yeah, like this one and this one. they're basically like placeholders for where a gene that will have like a phenotype can exist.
So how does that translate to the one with plus minus slash, like that thing?
So basically the slash is just the other allele. So if I wanted to talk about this person, it would be like, let's say like the gene is called, I don't know, like hair,
Mm-hmm.
like I don't know, hair BB,
Yeah.
and so this person like has, you know, the like dominant A as well, like dominant A, and then the recessive little a. So maybe I'd draw it like this.
Mm-hmm. Okay. Okay.
Yeah.
All right.
So for basically like, so for Fox oh, I wrote SPF. That's weird. SF, right? So this person, you know, if it we did like a Punnett square, one would be SF and the other would be like just whatever the wild type is. So yeah.
Okay.
So that's what that thing means. So like this guy, so this over here means that one allele is SF
Mm-hmm.
and the other allele is just wild type for FOXP3.
Mm-hmm. Okay. Got it.
Yeah. Okay. So okay, they're saying in a female mouse born with two wild type FOXP alleles, that means it's FOXP3 plus slash plus. Both of the alleles are wild type for FOXP3.
Mm-hmm.
Yeah. So okay, so that's what the mouse has, and they're asking what's the minimum number of inactivating recessive mutations that might be sufficient in the FOXP3 alleles in mammary epithelium to significantly increase the likelihood that this mouse will develop mammary tumors.
I guess I just have I kind of like didn't know where to even start to think about this question.
Mm-hmm. So if we know that 60% of those with SF as an allele and
Mm-hmm.
wild type FOXP3 allele produce
Yep.
mammary tumors, right,
Mm-hmm.
by age
Mm-hmm.
2,
Yep.
um, that's like more than 50%, right?
Yeah.
Wait, what?
No, it's just I mean like the phrase like significantly increase the likelihood, like I just like have no idea what they mean by that.
Where did you see? Oh, on the question?
Yeah.
So, what's the minimum number of inactivating recessive mutations that might be sufficient in the FOXP3 alleles in mammary epithelium um, to increase the likelihood that this mouse will develop mammary tumors? So, okay. What's the minimum number of SF alleles that this person can have to significantly increase the likelihood that this person will whatever develop mammary tumors?
Right. I guess the logical way to go about this is just by reading the passage, you just said that if it carries one inactivating allele, then it will develop, then 60% of them will develop too, right?
Mm-hmm.
So then it becomes minimally you need one.
Yeah, let's see if something else can help us, because the recessive part, right?
Yeah.
So I'm looking at let's see, the wild type FOXP 3 allele is carried on the inactivated X chromosome in 15% of non-cancerous mammary epithelial cells. but in 100% of cancerous mammary epithelial cells. And then above that, it says wild type FOXP3 transcripts are easily detected in non-cancerous mammary epithelium, right? From female SF and positive mice, but are undetectable in cancerous mammary epithelium from the same mice. So they're telling us that for mice that have the FOXP3SF allele along with the wild type allele, that it's 100% detectable. That is the wild type portion is 100% detectable in the ones that have cancer. But are completely undetectable in the ones that don't have cancer. So, let's see if I can see anything else here.
Also,
thing, yeah.
Does transcript mean like mRNA transcription? Okay.
Yeah. Where did you see that? Is that
the first bullet?
Oh.
The wild type.
Oh yeah.
Okay.
Yeah, transcripts. So basically, the FOXP3 like gene. Yeah.
Right.
So, okay. So and it codes for a transcription factor, it seems.
Yeah, it does.
Okay. So let's see. So all right. The question is saying 2 wild type FOXP3 alleles. Okay. So how many mutations that create scurfin? Okay, I think I understand what inactivating means now. I think it means that it inactivates the chromosome itself.
Yeah. Wait. Well, inactivating. Well, so in females, there's X and X, right? One X does get inactivated, just because, right?
To make sure that there's enough equal amounts of gene products in men and women.
Yeah, exactly.
Like a bar body.
Oh, okay.
Is that what you're talking about?
Yeah.
Okay.
But is it the same thing as the SF, the inactivating SF allele?
Yeah. I think I just made that connection now, because at first it just says inactivating, so I thought it would inactivate whatever gene, whatever FOXP3 does, and actually I'm still not sure, because,
So
you know what? Actually, never mind.
Yeah, I mean, it said, okay, so inactivating recessive mutation, does that mean inactivating is the same thing as recessive in this case?
Minimum number of inactivating. So, okay, so I think, all right, let me see. So, all right, actually, I don't think it has to do with a chromosome being inactivated. I think it just has to do with inactivating the
the allele
for the FOXP3 gene. But so wait, FOXP. Okay, so all right, okay. If I said that the FOXP3 gene works as a repressor and works against the tumor formation, okay, I get it. So it's like the opposite of what I initially thought. So the SF, the scurfin one. So basically, FOXP3 works against the creation of tumors and against the creation of cell death.
Yeah.
So Yeah, so when you have that the wild type replaced with something like SF, your rate of tumor or cancer and all that stuff
In-
increases
yeah.
substantially. Yeah.
Mhm.
So, hmm. So okay. Minimum number of inactivating recessive mutations. Recessive, I still don't really have a
Yeah.
complete answer for, other
Mhm.
than the fact that hmm, 100% of cancerous mam.
I mean, maybe it's just assuming that inactivating SF is recessive.
Yeah, I think so. I think they're asking just how many SF
Yeah.
is needed to significantly increase the likelihood. And I think so. I'm trying to see like what they mean by significantly increase. Like, hmm.
Yeah, exactly. I mean,
I mean, if
just
60%, if one SF leads to 60% of them getting tumors,
yeah.
and at one SF causes an 8 to 12 fold increase in herb B. Okay,
Hmm.
I think that's what gives us the answer. Because the
Hmm.
60%, I don't have like anything to like a control to
Yeah,
compare it
exactly.
to. But what I
Hmm.
do have something to compare is the herb thing.
Hmm. Yeah.
Because basically, okay, so I'll write this in red. So basically, herb B2 causes 8 to 12 times more, like, whatever,
Hmm.
cancer,
Yeah.
basically.
Yeah.
And, um, and then, if FOXP3 works as a repressor of that,
Hmm.
if you get rid of one FOXP3 allele, you'll have an increase in like an 8 to 12 fold increase, which is pretty significant, I suppose.
Hmm. Yeah, for sure.
So I think that's like the key here.
Yeah, for sure.
The
I mean,
60%, like, I guess that means that 40% don't, and if we're thinking of just greater than 50% as being significant, but I wasn't completely sure on that. But I think the herb B2 thing, that's 8 to 12 fold more, is the key here.
yeah, I think the herb B2 is more convincing, but the answer just said that it's based on the 60%
Let's see.
female.
Passage indicates that approximately 60%, which carry just one, get tumors. Therefore, the minimum number of inactivating mutations to increase the likelihood of tumor formation is one.
Yeah.
Um, yeah, I mean these answer these answer explanations always suck.
Yeah. I mean, I think what I was I mean, the best case scenario is just to go through what we did, like just look at the evidence based on the passage. But I guess when I was doing the question, I was confused because I'm like, what does it mean by significantly increase?
Yeah, yeah, yeah.
Because it's kind of so subjective. Because like
Yeah.
what if I think 10 to 8 to 12 times is not that significant? Because
Mm-hmm.
right, because it could be more than that. So it's like there's no way to tell. But the
Yeah,
best case here is probably just the
I mean, yeah. Like I'm still gonna go with the 8 to 12 because the 8 to 12% increase is just compared to is just like it's compared to like no. It's compared. I mean, it's compared to Fox P3 plus plus.
Mhm.
The 60% one, it's just telling us what happens when there's one SF allele.
Mhm.
It doesn't tell like what if Fox P3 plus plus is like 50%.
Yeah.
So
Exactly.
that's not enough. And these answer explanations always suck. So,
Mm-hmm.
like, so maybe I would like like if we weren't given 8 to 12 fold, I would use the 60%, but I would just compare it to whether it's less than or greater than 50%.
Yeah.
So since it's greater than 50%, aka the majority, need just one SF allele,
Mm-hmm.
that could be enough.
Yeah,
So
for sure. I mean, this question I think is just like weird.
yeah, it's weird. Yeah, for a couple of reasons. That one being like not having a control to compare it to, um, mixing up inactivating with
Mhm.
inactivated chromosome,
For sure.
not knowing what any of the recessive things
Mhm.
mean because
Exactly.
it doesn't say anything about recessive.
Right.
Yeah.
So you just have to go with it because it tells you that.
Yeah, I think what helped the most is just knowing that FOXP3 like inhibits cancer, and the introduction of, or just replacing it with something else, um, causes like cancer at least 8 to 12 fold more, and then ones that have that replaced, it's 60% of them, make cancer. So yeah,
Mhm.
what did you end up choosing? Because the
I
recessive part makes me also think of like two as an answer. Because if it's recessive, but whatever.
no, I guess I have no idea. I mean, it's a multiple choice. I just pick one.
Mm-hmm.
I have no idea, but
Mm-hmm.
makes sense. Okay. I mean, I guess we can. Yeah, I think we can leave the rest for next time probably.
What the?
Yeah, like the chem phys section.
Yeah. How long did you want to do today? Because I have an hour 30. It says this is going on for an hour 38.
Oh,
So if you wanted to do like 2 hours,
okay.
we could do like the rest of the 20 minutes. What do you want to do?
Okay, yeah. Okay, yeah, yeah. Then we can do
The
maybe two
CP?
more questions. Yeah, the questions I sent they're mostly discrete, basically, so we can go over it.
Yeah, CP is like usually
Yeah, exactly.
like that.
Yeah. Oh, one more question
Yeah.
I had about, I was gonna ask you about the BB section is, so through like my practice and everything, I think my score hovers around like 128, 129. And so I usually get about like 10 questions wrong.
Yeah.
And I'm just not sure like how to improve on that score.
Yeah. Have you noticed any patterns in those 10 questions that you get wrong?
Yeah, it's mostly from the genes and the cell biology part.
Okay.
I don't know.
So, yeah, yeah, if you can show me like, more of those. But so far it seems like we've made some progress in terms of reading our like charts and graphs and stuff.
Mm-hmm.
Like
Yeah.
how much would you say that is an aspect of the things that you get wrong?
Um, I mean, they don't really ask about that many
Mm-hmm.
figure questions. Like usually, if they have a figure in a passage, they'll have one question on it or like two most.
Yeah, yeah.
So I feel like I was able to get past it, but so
Yeah.
I don't know.
Okay.
I think sometimes it's mostly like just like reading and like understanding the
Mm-hmm.
passage the correct way that they want you to understand it.
Mm-hmm.
Or like sometimes it could be so usually okay, so I remember that out of like the 10 questions that I get wrong, for example, 3 to 4 would be like just dumb mistakes that I just like didn't think properly or like it's very stupid. So I don't know what to do.
Uh-huh. Yeah, so far it seems like, let's see, one, two, three, four. Seems like 4 out of the 7 that you sent me had something to do with the chart or the figure?
Mm-hmm.
Um, so that's just like what I've seen so far. So yeah, so if we can go through like more passages and be able to like write things out in shorthand and see like exactly what's meant,
Mm-hmm.
that could help. But
Yeah.
yeah, I need like more data.
Yeah, no, for sure, for sure. I think
um,
I'll
yeah.
go through some of the other practices and just get those questions maybe.
Mm-hmm.
Okay,
Um,
sounds good.
have you noticed any like patterns in any other sections?
Uh,
Well, okay, let's do the CP one. So, I see
okay.
this thing. It's about the Haber production process.
Oh, I
Uh,
was thinking
yeah.
we can go through passage 10 because those are more like discrete questions.
You mean the ones that aren't passage 10 or more discrete?
I mean, for those ones, I think it's easier to improve on.
Okay,
But
so you want to do passage 10 because they're more discrete,
I guess,
or
yeah.
or the opposite? Okay.
I mean, you know what? No, no, no. You know what? Let's just do the passage one first. Let's just do passage six first.
Okay. So, it is possible to design a reactor where the SCY um, conductor. Okay, that seems to be this ceramic strontia. Okay, I'll take a look at that later. And the nitrogen ammonia electrode operate at different temperatures. Which combination of temperatures is expected to give the best results? Okay, so let's see. Born-Haber process, okay. Exothermic. High temperature favors reverse, okay. However, high temperatures are required to achieve a reaction rate fast enough for industrial use. Okay. Despite these limitations, alright. Um, however, okay. They need a catalyst, okay? So electrolytic cells can be used to produce ammonia. In one such method, hydrogen is pumped into one compartment of a reactor constructed of SCY. Okay? So that involves that involves hydrogen turning into protons. Okay, the protons are transported through the solid SCY electrolyte. Okay, I think they meant electrode maybe, but okay. Let's see, to react with nitrogen at the other electrode, according to half reaction 3. Okay. All right. SCY conductors are favored for this use because their proton conductivities increase substantially with temperature. All right. So I'm going to start to write some stuff down here. So let's see. So okay, let's see. Um, so so first I'll just write something like this.
Oh,
What's up?
wait. Can you repeat? Uh, so wait. So you said that high temperature, the reverse reaction was favored?
Yeah.
Wait, where where did you see that? Oh
Um,
wait, it's from the exothermic part.
yeah.
Okay, okay. Yeah. Hydrogen is so that's the second part, right?
Mm-hmm. Yeah, the second one is so first one is the Haber process, and the second one is an electrolytic process.
Mm-hmm. So I guess I was very confused about what electrode they were talking about
Mm-hmm.
because it doesn't like say electrodes. I wasn't sure. So what? So why are they comparing SCY temperature with the electrode temperature?
Um, Wait, let me just finish this. Okay,
Oh,
both
yeah. Yes.
cases upper. Okay, I got it. So, all right. So basically, um, so well, the Haber process information's here. Like, it works at 450 degrees Celsius, but the higher it gets, it's gonna go in the reverse direction. But it's kept in the forward direction through high pressure, which is 15 to 30 atm.
Mm-hmm.
Also through using a catalyst.
Mm-hmm.
The other process involves H2 gas being thrown into an SCY like electrode, which creates like protons essentially, which enter into another electrode to interact with nitrogen gas to produce ammonia.
Mm-hmm.
Um, so um, in both, oh, and then the SCY electrode, uh, its proton conductivity increases substantially with pressure. So I crossed out the high T there, and I'll put it here.
Mm-hmm.
Now, now, oh, and then for both cases, if you're less than 300 degrees Celsius, it's good, right?
Mm.
But anything greater than that causes like this reversible decomposition of ammonia, which basically is against what we're trying to do, which is to create ammonia.
Mm.
Okay, so they're saying you could design a reactor where this SCY conductor and the nitrogen ammonia electrode, which is this one over here, operate at different temperatures. So which combination of temperatures is expected to give the best results? So only the SCY like part needs to be at a high temperature.
Yeah.
So, this, if you can make it so this is high temperature and this isn't, like that would work. Because overall you don't want the temperature to be that high.
Right.
And since you know that the SCY part works at high temperature, but overall you don't want it to be that high, you could just make it so only that part has a high temperature.
Wait, yes. Okay, I see now. So how did you draw the second box? Like, where is it? Where does it say?
What's up?
So where did you get the information for the second box?
Oh, that's just so this is the SCY like conductor or whatever. And this is like the electrode. So so they're saying that in the passage for half reaction 2, that's what happens in the SCY part.
Mm-hmm.
And then it's transported to react with the nitrogen at the other electrode, and that's half reaction 3.
Oh, okay.
And then it says SCY conductors are favored for this use because their proton conductivities increase substantially with temperature.
Okay, so the electrode they're saying in the answer correspond to the solid SCY electrolyte, right? That's what they're saying?
So I don't know if they made a typo or not, but I think they mean they mean that there's only one electrode, and that's this one.
Okay, I see.
The other one, like they call it an electrolyte, or they call it like a conductor, and in the answer choices they're saying higher than electrode temperature, so they both can't be electrodes. So I think the electrode is just the second like box.
Okay, I see. Okay.
Yeah. So basically, like, they both require like whatever high temperatures, but it gets bad when it's high temperature. So if you can just make the conductor part high temperature, then we can, we don't have to get into the bad, like into something that's too bad, where it's gonna start the decomposition of the ammonia.
Yeah, I agree. Okay, I see. It's just electrode wasn't mentioned in the passage, so I wasn't.
Yeah, it's mentioned in the where is it? At the they say other electrode, so I guess they're both electrodes, but like right under half reaction 2 it says the electrolytically generated protons are transported through the solid SCY electrolyte to react with the nitrogen at the other electrode.
Yeah. So yeah. So I guess they're both electrodes.
Yeah.
Yeah. I guess I was just having a hard time visualizing what the process looks like. So yeah.
Does it help like the way I drew it?
Yeah, for sure. But I'm just not sure if I could independently come to that conclusion.
I think you can, if you're able to like, separate it in these ways. And I'll like copy and paste this into the notebook for you.
Okay. Yeah. Sounds good. Okay. Let's see. Maybe we can do one more question.
All right, the glass rod one. Or yeah, because I think the other ones will require me to look at the passage, which might take longer.
Sure.
I mean, it's up to you.
Yeah, no, we can do the glass rod thing.
Okay, so it's rubbed with a silk scarf, producing a charge of that. And just know that the magnitude of the proton electron charges is also that, or is the 1.61.
Mm-hmm.
Okay, so the glass rod has. Okay, so how many things like added to it, right?
Mm-hmm.
Okay, so it seems like it starts off like neutral, right?
Mm-hmm.
Okay, so basically, we're just going to divide its charge by, the amount, like the 1.61.
Mm-hmm.
And by the way, you won't ever see protons added or removed unless it's like a fission or some type of nuclear reaction. So it's always going to be electrons.
So that's where it went wrong because I saw that the charge went positive, so I was like, oh, okay, protons.
Mm-hmm.
But
Yeah. So yeah.
But then I was confused because I thought you could add protons. But like, why can't you add protons?
Oh, just because they're in the nucleus?
Okay, so right. So does that mean only electrons can pretty much freely move around?
Yep. Yeah, I
Okay.
usually say like chemistry is like the study of electrons because electrons move around, right? Like they're the things that move.
Mhm.
The protons, like they define the identity of the element. They're in the nucleus.
Yeah.
So unless there's some type of atomic, reaction like fission or fusion,
Yeah.
like protons, nothing, like like you can't get that deep into an atom.
Yeah. No, fair enough. Fair enough.
Mm-hmm.
Because I was just confused because
Mm-hmm.
because removing electron will also give you a positive charge because you're removing a negative charge.
Mm-hmm.
Okay. I just didn't think.
Yeah. So for this one, you could even, well, yeah, you can first cross out A and C, I suppose.
Yeah.
And then you would have to do some math.
Yeah. I definitely did math first. So,
Mm-hmm.
yeah. Okay, I see. Makes sense. All right. I think that's it. We can leave the rest till next time, I think.
Okay.
Well, I could ask you another question. So,
Sure.
I in the chemistry and physics section.
Yeah.
So I'm also very weak in organic chemistry.
Mm-hmm.
And it just happened that the day of my exam, I didn't really get any organic chemistry questions.
Mm-hmm.
So I definitely lucked out there. But I think my understanding of organic chemistry is like very, very basic.
Mm-hmm.
And do you think that's enough to answer the questions on the MCAT?
So I'll send you some stuff that I guess I haven't sent you yet, like from my other notebooks where I talk about organic chemistry, for instance. Yeah, like I have something where I talk about, I talk about organic chemistry, and I sum it up by saying how electrophiles and nucleophiles react.
Mm-hmm.
And
Yeah.
so I'll send that to you. I'll put that in your notebook.
Okay.
Yeah, I'll put like a bunch of things in your notebook now that you
Okay.
can look over. Okay?
Yeah, I mean, it's just for organic chemistry, I think I was able to get around it by just like knowing some of the key concepts,
Mm-hmm.
by like not really learning it because I didn't learn it
Yeah.
in school. But I was able to just kind of like figure it out. But like sometimes if they ask complicated reactions, then like I get stuck.
Yeah. If you come across anything, send them to me, and I can like help you through it, like help you like
Okay.
know like what to know and what not to know. But I think when I did like the math, there's only like, let's see, there's only a total of let's see, because it's organic chemistry is 20% of the total exam, with 15% in the CP section and 5% in the BB section. So that comes out to 8.85 for CP and 2.95. So roughly, like, I guess 12 Orgo questions total.
Yeah, I mean, I think Orgo, I mean, like, so if they ask about like RNS and whatever, that part is okay. Like, I can get
The
those
what
parts.
stuff?
Like, if they ask questions about like, oh, what's is this E or Z or is it R or S? Like,
Oh yeah.
that kind of stuff, like, I'm fine with that. But it's
Oh,
just
they'll probably ask you stuff like that. That could be annoying sometimes. Like the questions I've seen have been like, yeah, I've been asking about like, I guess like a number of chiral centers.
Mhm.
Is this reaction going to produce a racemic mix, like a racemic thing, or something like that?
SMO.
Yeah, like maybe some questions about like maybe like some compound, and then you add like this reagent followed by this reagent, and what kind of thing would you end up with?
Yeah, yeah, predicting products definitely I'm not very good at, but
Yeah. So those could be kind of like tricky. Um,
mhm.
so yeah, whatever you come across, like send to me. Like the more questions, the better. So I
Mhm.
can, have like a better idea of exactly what type of questions you need, what type of problems you need, uh, like work on.
Mhm. So I think for next time, because I haven't done any AMC since, so we can do. I did blueprint questions,
Oh yeah.
so
So those are really
maybe
good. Like yeah, those are really really good. Because they're harder than the AMC stuff.
mhm.
Yeah, and yeah, when I did like the spreadsheet thing of like people who scored 100th percentile, like they they had like blueprint scores like 12 points lower than their actual scores. So blueprint stuff is really helpful. Like, what have you been doing for blueprint?
in, in terms of score.
Oh, like yeah, like what have you been doing on the blueprint like website? Have you been doing like the practice exams?
Yeah, I've just been doing the practice exams.
Yeah. So you don't need to do the cars for blueprint, and you don't really need to do the PS either. But the CP and the BB is very useful on blueprint because they're going to be harder than what the AMC throws at you. So they're just really really good practice. Like
Mm-hmm.
so yeah. Have you been like what have you been doing on that?
How have it been?
Oh,
How
what have you been doing on the blueprint stuff?
just well, I've done most of them except for full length 8 to 10.
Oh wow! So you've done like 1 through 8
Oh,
or
one
one
through
through
seven.
seven?
Yeah, because there was this for 1 through 7 was from my previous studying session, like my when I studied for
Yeah.
it before. So yeah,
So,
so I
uh-huh.
yeah, because I have 3 more weeks until my exam. I was thinking I could do the last three just like every week. And
Yeah. Yeah, and if, you want to like redo those, I mean, yeah. How were the CP and BB sections for the first 7 that you did?
I think some are hard, some are okay.
Okay. Yeah, they're all basically like harder than the AAMC stuff.
Yeah, definitely.
Like people who scored like an average of like 524 point something on the actual test scored like 511, 512 on the at next step stuff. So they're a lot more science heavy, calculation heavy.
For sure.
So if you can go through those and find questions, that is also going to be helpful. Like maybe like if you have some time, like going through like what you answered for the tests 1 through 7, and like going just I guess like go through it like quickly I guess, and then if you see a question that you don't like that doesn't make sense even after looking at the explanation and stuff, Yeah, if you can send me that kind of material, or maybe do like practice test 8, or at least just the CP and BB sections, and then sending me like questions that you have from that could also be like helpful. Yeah.
Okay,
But
sounds good.
yeah, that material is really good.
Okay, sounds good. Yeah, I agree. But yeah, I agree. So I think even if it's harder, I feel like the real exam is not easy. So I think it's
Yeah.
probably okay.
So I think it's more like if you're able to do like the harder material on the blueprint exams, that when you come across something that's going to be, like whatever, out of the blue, no pun intended,
Mhm.
it's going to be you're going to be better prepared having done the blueprint stuff.
Okay,
Just
sounds
because
good.
like AMC, like there just isn't enough material. Like there's
Which
4 practice tests, and there's like the section banks that everyone has done. So in
Mhm.
order for them to make an MCAT that has good like viability or validity or whatever, I forgot what it's called in PS, but like in order for it to be a good test, right?
Yeah.
You're going to need it to, yeah, you're gonna need to have it be very different from what everyone else has studied already. So if yeah, I think the blueprint stuff will help put you like you know, on like a higher
Okay.
level. Yeah.
Okay, sounds good. Do you have time tomorrow?
Yeah. What time works for you?
Anytime.
Okay. Okay. Let's see.
Yeah.
So tomorrow I have something from 4:45 to 5:30, but we can do like before. We could do after. What do you want to
Hold
do?
on. Wait, 4:45. Wait. Sorry. What did you say? It was what time
So
does your
that for
thing end?
you, I guess, would be like what, like 1 or 12:45?
Wait, are you
Twelve
saying
forty-five, I
before
think.
one or?
Oh, so just basically, I just have like something from 4:45 to 5:30, like my time zone. So it could be before that or after that. I think before
Okay.
that would be best or something. So
Right. So
let's
how
yeah.
much before that can you do? If
Um.
you do the mornings,
Uh.
what for you would be like early afternoon?
Yeah. Yeah. So let's see. Like, okay. So what were you thinking?
I can do either really early. Well, for me, I can start at 7 a.m. here, which is like 10.
Yeah. Yeah.
So like
Yeah.
I can, yeah, 7 a.m. is like where I could start.
Okay.
So,
Let's.
but then I have something at noon, which is
Mm-hmm.
um 3 p.m. for you. So anytime before that could
Oh.
work.
Okay. Okay. Um. So let's see. Maybe like 1 p.m.
So 1 p.m. is like 10. No, 9. Uh, yeah. So it's 9 a.m. here for me, basically. That could work, but I could. Yeah. Okay. So if we start at 1 p.m., then I can do 2 hours.
Yeah.
And then I think we could go over. I did one section of the blueprint campus, so I can go through the questions tonight just to see which ones that we can work on, and then we can do those tomorrow.
Okay.
Does that sound good?
Yep.
Do you have access to the blueprint, or do you want me to screenshot?
Um, I have access, but I don't think I did. Wait, let me see. Blueprint. Which full length are they from?
Well, I was thinking of the full length 7 because that's the most recent one that I did, but
Okay.
um,
Oh well, I did it for the. I have the ones for the short exam.
oh, oh,
So let's see. That means that. Oh, you know what? I can't. I can't see it. I just tried.
it's okay. I can just send it to you. It's okay.
Okay.
Yeah, I can just like put it in a package and then send it to you, and then we can go over it.
Okay.
Okay, sounds good. Yeah. Okay, sounds good. Oh, by the way,
Yeah.
um, are you applying to med?
I mean, like